Bismillahirrahmanirrahim. My name is Anisa Zebawan and I will discuss the biofortification of rice with carotenoid and reduction in cadmium accumulation. Carotenoid biofortification. For the biofortification of rice with carotenoids, CRISPR-Cas9 technology can be used. Beta carotene is an important type of carotenoid and is the precursor of vitamin A. Biofortification with beta carotene may help in preventing blindness and improving the immune system. Golden rice is considered one of the important source of carotenoids. So one of the experiment to increase the carotenoid accumulation in rice callus was by inducing the OR mutation. So to enhance the concentration of beta carotene in rice crops, an attempt is made in 2019 on rice callus. They modified the OSOR gene using CRISPR-Cas9 technology for the accumulation of beta carotene in rice callus. This research idea is inspired by the observation of cauliflower orange mutant, which is orange in color due to high level of beta carotene in its non-photosynthetic tissues. So the gene which is responsible for high beta carotene content in cauliflower is the OR gene, and the ortholog of this gene is also present in rice. So the orthologs are the genes whose functions have been changed during the evolution process. So if we induce the mutation in OR gene present in the rice, then we can also achieve the high beta carotene level in rice. So if we look at the explanation of this procedure, then the directed disruption of the OR gene using CRISPR-Cas9 in rice can produce carotenoid accumulation phenotype in rice. The OR gene codes for OR proteins that play a significant role in carotenoid biosynthesis. The callus is selected for this experiment because we can detect the change in color within less time after carotenoid modification. The two ortholog genes in rice are OS02G0651300 and OS02G053500. The OS02G0651300 is selected for the a mutation because its expression in calli system was higher than the other gene. This gene is named as OSOR. A single guide RNA is designed to mimic the OR mutant of cauliflower. CRISPR-Cas9 is used to induce sgRNA guided mutagenesis in the OSOR gene by deleting the sequence between the third exon and third intron in the OSOR gene. The orange color carotenoids are extracted from the rice calli and analyzed using high performance liquid chromatography. The carotenoid that was detected in the rice callus was beta carotene. This phenomena is not used in field still, but this phenomena can be used in practical after refinement and further analysis. So here is the whole procedure of this this phenomena, in which a guide RNA is designed that is along with the Cas9 protein is allowed to transfect the calli cells. This transfection is mediated with the help of Agrobacterium tumefaciens. So three different kinds of guide RNA were designed: OSOR T3, OSOR T1 and OSOR T2, and these three guide RNA will target different positions between between the area of exon 3 and intron 3. So the best mutation was induced by OSOR T3 when the mutant calli cells were selected with the help of hygromycin. Then it was observed that best mutation is carried out with the help of OSOR T3 guide RNA. Then is the low cadmium rice. Before creating healthier varieties of rice, one procedure is to eliminate cadmium toxicity. Cadmium is a toxic metal found in rice and poses serious health threats such as cardiovascular diseases, osteoporosis, cancer, etc. According to a report, Japanese people consume 80% of cadmium from rice. Its concentration is higher in indica cultivars as compared to japonica rice cultivars. So to reduce the accumulation of cadmium, CRISPR-Cas9 can be used to knock out the metal transporter gene. So one of the experiment was the editing of OSNRAMP5 OSNRAM5 gene for low cadmium accumulation in rice using CRISPR-Cas9. Cadmium is majorly accumulated in rice through its uptake from the soil. A natural resistance associated macrophage protein OSNRAM5 helps the rice to absorb cadmium. So if the mutation is induced in OSNRAM5 gene, it may cause decreased cadmium accumulation in rice. Then 3 knockout mutants of OSNRAM5 in 2 Japonica varieties are created using CRISPR-Cas9 technology, and it was observed that in all 3 mutants there was a reduce in cadmium accumulation in the all tissues. The OSNRAM, but the problem is that the OSNRAM5 protein also play a role in manganese uptake. So there was a probability that its knockout may affect the plant growth due to low manganese. When the growth of mutants is compared with the wild type, a growth of mutant types was gently decreased at low manganese concentration. And also the agronomic traits such as plant height and the productivity or yield were decreased in OSNRAM5 mutants, but this reduction was mild, about 20%. So in this study it was argued that Tang et al. In 2017 also performed the same experiment in which he knocked out the OSNRAM5 in indica cultivars that resulted in low cadmium accumulation without compromising the yield. So they argued that the high soil pH in their fields and heavy water drainage may be the cause of manganese deficiency, and it is concluded that different growth conditions may have different effects on agronomic traits of rice with OSNRAM5 mutation. So here is the procedure of this mechanism in which a guide RNA is allowed to transfect the rice cells. So here actually the 3 kinds of guide RNA were formed by deleting the base pair at 3 different positions. So here are the references. Thank you.